Good morning to everyone. My name is Alícia Martinez-Varela and I am a PhD candidate at the Environmental Assessment and Water Research Institute from CSIC in Barcelona. And I'm here to present an oral for the session Biodegradation of Organic Trace Pollutants in the Environment at the SETAC Europe 2021. The presentation is titled Particle-associated bacteria in sea surface microlayer is the major player in pH biodegradation in coastal Antarctica. This being said, let's get started. Polycyclic aromatic hydrocarbons from now on PAHs mostly originate from incomplete combustion of fossil fuels and biomass, and to a lesser extent from well spills and natural processes such as oil seepage. Many of them are hydrophobic, bioaccumulative, and toxic, posing a threat to ecosystems. Their semi-volatile nature allows them to be transported in the atmosphere, and in consequence they are ubiquitous in the global ocean, even reaching remote polar regions far from their source points. Quantification of atmospherically deposited pHs into the ocean estimate a global monthly entry of 0.09 teragrams. And you're probably wondering, what would that mean in terms of carbon? It would equal to having 4 deepwater horizon oil spills accidents every month in the global ocean. In the oceans, the main collector of atmospherically deposited particles and compounds, including PAHs, is the sea surface microlayer, from now on SML. The SML is the thin interface between the ocean and the atmosphere, where particle and gas exchange between the ocean and atmosphere happens. Under favorable meteorological conditions, the SML physicochemical and biological features can largely differ from those at the subsurface layer, from now on SSL. The active air-sea exchange and mixing dynamics of particles, gas and water between the adjacent environments contributes to the enrichment of amphiphilic, hydrophobic, and an array of miscellaneous organic molecules at the SML. Bacteria inhabiting the SML are known as Bacterio-Neuston and are exposed to unique physicochemical properties and harsh environmental stresses. Bacterio-Neuston is believed to participate in the mediation of air-sea exchange interaction and key biogeochemical and climate-related processes. In the Southern Ocean, atmospheric diffusive loadings and snow deposition represent the major entry source of pHs in the ocean. As observed in various oceanic regions, pHs accumulate at the Antarctic SML due to their hydrophobic nature, with total enrichment factors up to 8. Recent flux assessments reveal that only 1% of the total airborne pHs are sequestered to the depths by the biological pump, suggesting a major fraction of aromatic hydrocarbons is removed at the surface ocean by microbial degradation and minorly by other abiotic processes. Despite the key role of biodegradation, our understanding of the microbial processes eliminating aromatic hydrocarbons from the upper ocean is very incomplete, even less for the SML. The goal of this study was to measure under experimental conditions the pH biodegradation rates, along with characterization of bacterial communities at the SML and SSL in coastal Antarctica, and the responses of SML and SSL differentiated marine microbial communities and size fractions to environmentally relevant concentrations of pHs as an anthropogenic dissolved organic carbon model family. We conducted the experiment at the Antarctic Spanish base Juan Carlos I in Livingston Island in the South Shetland archipelago in Antarctica. We conducted the seawater sampling in February 21, 2018 at South Bay using a glass plate sampler to collect the SML, as you can see my colleague Gemma and I are doing in the photo. We challenged Bacterio-Neuston and bacterioplankton with background concentrations of pH. This is 200 nanograms per liter. These concentrations are in the range of the natural variability of pHs in the surface ocean. We incubated the experiment at in-situ temperature and in the dark over 24 hours. Non-spy water was used as experimental controls, while HPLC spiked samples were run as abiotic controls. We quantified the pH concentrations over the course of the experiment and assessed the responses for size-fractionated, free-living, and particle-associated microbial communities at functional and structural level by means of metatranscriptomics and 16S rRNA amplicon sequencing. Results and discussion section, we start with the pH removal rates. Note that on the left we have the experimental control with the y-axis from 1 to -1, while on the right there are the SPECT samples with a y-axis that goes from 1 to -4. Dots below the zero line indicate removal. The x-axis are the octanol water partition coefficient for the pHs measured in this experiment, so the more to the right, the more hydrophobic the compound is. In blue, those pHs with low molecular weight, these are those ones with less than 3 fused rings and less hydrophobic. And in red, the high molecular weight pHs, those with more than 3 fused rings and thus more hydrophobic. Interestingly, we observed significantly higher pH removal rates in the pH treatments of the SML than in the SSL, in particularly significantly higher removal rates of individual high molecular weight pH compounds. This can be due to high molecular weight pH hydrophobicity facilitates absorption of these compounds to suspended particles, expectedly more abundant at the SML. However, results from the abiotic control show that partitioning of pH to cells, phyto, and bacteria can account for a decrease of concentrations of 10%. So, this points that removal of high molecular weight pHs has a biogenic origin. By means of 16S rRNA gene sequencing, we characterized microbial populations for each fraction. This is free-living bacteria and particle-associated bacteria. We compared the relative abundances of taxa between control and the pH-amended water to assess the response after 24 hours of pH exposure in each layer. In the SML, a significant notable growth of all the genera groups known to be able to degrade pH was observed in pH-exposed communities. In particular, what were the taxa increasing substantially? Well, the UV-resistant and facultative pH degrader Pseudohalteromonas, which increased the relative abundance between control and pH treatment with fold changes of 7 and 13 in the free-living and particle-associated cohorts, respectively. Pseudohalteromonas increases suggest even subtle pH concentrations can trigger the exponential growth of low-abundant taxa at the SML. This low-abundant taxa is known as the rare biosphere, becoming key players for pH interaction as previously observed in the Mediterranean all-polluted seawater with other hydrocarbon-degrading genera. In fact, Pseudohalteromonas' nutritional preferences towards pH have been described in several studies, appearing abundant and as early responders following marine oil spills, as well as pH-enriched mesocosms experiments at low temperatures. Moreover, Pseudohalteromonas, among other alteromonadales, such as halomonas and alteromonas, have been reported to produce exopolymeric polysaccharides, also known as EPS, with amphiphilic properties. Surfactant-like EPS increase hydrocarbons solubility and bioavailability by increasing the interfacial surface between pH and other hydrophobic substrates and bacteria, as well as providing a physical structure, facilitating faster transformation steps. So, as we see growth at the SML, was this reflected at community functional level? The activities of the different groups of bacteria 24 hours after pH exposure were assessed with the metatranscriptomes. In the figure, you can see the degradation pathway of the simplest PAH, naphthalene, and the heat map of fold change between control and pH exposed samples for the pH degradation genes obtained after performing a hammer search with the selected PFAM profiles. The ring hydroxylating dioxygenase, the enzyme initiating the degradation pathway, was found enriched in all pH-exposed communities except for the SML particle-associated. In fact, at the SML particle-associated fraction, most of the pH-degrading genes were found downregulated at the pH-exposed community. This information, coupled with the 16S community composition fingerprint showing the highest number of community shift responses led by Pseudohalteromonas rapid responder, suggests the peak of degradation was surpassed after 24 hours of pH exposure at the SML particle-associated fraction. The opposite story was observed at the SSL particle-associated, with the whole battery of pH-degrading genes upregulated. while the free-living fraction of both layers was in an intermediate stage. Altogether, our data suggests that each fraction and layer interacted at different paces with PAHs. Conclusions. Significant higher removal rates were observed for the heavy molecular weight pHs at the SML pH treatments compared to the SSL. Taxa related to pH degradation significantly increased the relative contribution at the SML, especially at the particle-associated community. Relative abundances of pseudoalteromonas increased the relative contribution exponentially in both bacterial fractions from low-abundant taxa to dominant taxa, suggesting pseudoalteromonas played a key role in pH degradation at the SML. pH-degrading transcripts were depleted in the SML particle-associated microbiome in the pH treatments after 24 hours, after significant changes in community structure had occurred, suggesting the most pseudometabolic potential to degrade pHs at environmentally relevant concentrations was the SML particle-associated microbiome. Our findings provide experimental evidence that pH at environmentally relevant concentrations at the SML is another, although neglected, community composition driver. Before finishing, let me introduce to you the Global Change in Genomic Biogeochemistry Research Group, because as you know, everything is about teamwork, so here are all the team members, and also you can follow us on Twitter if you're interested in following our news and latest publications. And finally, thank you for your attention. Special thanks to the organization of SETAC for adapting the conference 2021 to the COVID circumstances. And now I'm open to answer any question you have regarding my presentation. Thank you.